I'm going to talk about autoimmunity, autoreactive process in immunity, and So, in our lab, we are interested in type 1 diabetes, which is a T-cell mediated autoreactive disease. In a normal person, you see here a pancreas slide where we have stained the islet, the beta cells of the pancreatic islets for insulin. In a type 1 diabetes patient, insulitis can lead to an almost disappearance of the islet cells. This disease is mediated by T cells, as illustrated here. Again, an islet stained for insulin, and a part of the islet is depleted of insulin. And this part has been attacked by CD8 cells, and mostly of CD8 excitotoxic T cells. The CD8 or CD4 T cells are actually recognizing autoantigens specific to the islets of the beta cell on the islets, and their way progressing to the autoreactivity. This shows you the natural history of type 1 diabetes. In a normal person, you have the beta cell mass, which is present on 100%, and as you all might know, type 1 diabetes is a disease with a strong genetic compound. And when inciting events that can be from the environment, from the diet, or also from viral attack, or in starting the insulitis process, the mass of the beta cells decline. And the only way at the moment to measure this decline of a beta cell mass or the insulitis is by measuring autoantibodies in the serum of patients for some typical autoantigens. This is the only way to diagnose the disease onset. But we know now that the insulitis and the autoreactive attack is occurring already before the starting of the decline of the beta cell mass. And the main actors are the T cells. And that's therefore we are very much interested in better understanding those T cells and being able to phenotype them and characterize them. First of all, to provide a better diagnostic way, and secondly also to provide new ways of treatment. Here you see that the seroconversion, so the beginning of production of islet antibodies in young patients, has two very clear time scales. So there is one period at the age between 9 months and 2 years where most of the children start to seroconvert, and there is another rare period before this age at half year. This data comes from the baby diet study, which has been conducted by the laboratory of Annette Ziegler in Munich. This is a unique collection of samples from children that have been followed from birth and which have a higher genetic risk for diabetes. And so this collection of samples is a very good opportunity to look at autoreactive samples at the very early stage. So this very early stage is the stage of interest if we want to look at these autoreactive T cells. So we have the opportunity to have access to all these samples from all these children with genetic risk, which some of them will progress to autoreactivity, some not. And here you can see one example of one of these children. Here you see the seroconversion profile of one of those children, which first develops one autoantibody, then another one, and then another one. And a child with 3 autoantibodies has a very, very high risk and will probably proceed to open diabetes. So the samples we collect that have been collected and we have access to include samples before seroconversion. So they have no autoantibodies at this time, and then samples progressing throughout seroconversion. And what we want to look at is at islet autoantigen responsive cells in those samples for the typical autoantigens from the islets, but also from responding to other, as a control to other autoantigens as tetanus toxoid, for example. So what we do with the samples, we obtain the samples from different time points, and then we interrogate them for responsiveness to this typical autoantigens. So what we do, we take PBMCs we isolate PBMCs from the samples and incubate them with autoantigen for 5 days, while staining them for CFAC, so that we can select then autoantigen responding cells after 5 days. Here you see in a child that remains autoantibody negative, we have almost no cells that have been proliferating, whereas in a positive child, we can nicely select for cells that have been proliferating. Those cells are then single cell sorters, responder cells. When we look at the quantity of cells isolated in children that are autoantibody positive or negative, we can see that there is a clear difference. This is just plotting the cells from various samples, from 10 positive children and 8 negative children, and looking at the CFC superDIMs, or cells that have been clearly proliferating, and that are CD25 high and memory cells, comparing both, and you see clearly there's a quantitative difference. So we asked whether there's also qualitative difference. That's when we turn to doing gene expression profiles by looking at the single cells that we had sorted. So our strategy is, as already said, we do the stimulation assays for specific autoantigen. We isolate the cells directly into PCR plates. We do reverse transcription. We do preamplification with the primers of interest, also including TCR primers, to be able to follow up TCR clones if we want. And then we go onto the biomark and draw our gene expression profiles. Or alternatively, we take the same amplified DNA and we do Sanger sequencing of alpha and beta chains of the T-cell receptor, so that allows us to follow up clones. In the instance of those children, this was not making much sense, because the the repertoire of those children is extremely large, so it makes it very difficult to follow up clones. But in adults, we can do it very well. So after taking those cells specific for the autoantigen responding cells, we did just a simple a clustering of all the cells responding to one specific autoantigen. And what we can see is that the cluster composition is very different between autoantibody positive and autoantibody negative children. Highlighted is here, for example, a cluster that is specific to the autoantibody positive children, which is an interferon gamma specific cluster, whereas this cluster is almost absent in the negative children, and those children have a very high representation of FOXP3 cells, which are specific of T regulatory cells. So this showed us already as a first thing that those that the cells can have different phenotypes if they are coming from islet positive, islet autoantibody positive or negative cells. So here you see just a heat map looking at the sample pre-seroconversion. So at the moment where we still don't know whether the children are going to be developing the diabetes or not. The children are, those children are all very young of age, so half a year old, and we draw the gene expression profiles just of this time point. You can see here in blue are the autoantibody negative children, in red the positive children, and what is very apparent here is that the positive children are devoid or mostly devoid of for this FOXP3 cell type, which is the T regulatory cell types, whereas again some cell types appear more prominent in the children that proceed to seroconversion. Those children express TBET, which is a transcription factor for Th1 or interferon gamma producing cells, and also EGR2, which also is a transcription factor. So this is very simple analysis, but it showed us that we can already discriminate between children that will progress to autoreactivity and children which will not at the age before we have measured any autoantibodies, which is making us believe that we maybe can use this kind of analysis as biomarkers in the future. Now I just show you the progression of the disease in two exemplary children, which have been seroconverted later. So again, you have the very early samples, and then throughout the age of the children, the development of different cell types, as we can see here. So in blue, there is seroconversion. This child has seroconverted sometimes between the sample taken at the very early age and 1.2 years. And as you can see here, between this time and this time, we have a subset of cells which is appearing, and which is this interferon gamma producing cells, the same cells that had been that we found enriched in children with auto antibody positivity. The similar thing is observed in the other child, where we see also this appearance of these interferon gamma cell clusters over time. So this again shows us that this profiling of single cells in those children helps us to follow up the pathogenicity of the disease. Now, what kind of cells these are, we are still currently under, we're still trying to find out. We are also using different FACS-sorted cells to define phenotypes. One thing we did, we had also looked for cells that were activated with tetanus toxoid, which is also a Th1 specific, which also elicits a Th1 specific response, which is also similar to the interferon gamma response we see here. And we were just asking whether these two responses were similar. This is the last thing I'm going to show. Those responses are not similar. You can see here tetanus toxoid responding cells have a large interferon gamma responding cell cluster, but with concomitant expression of IL-21, which is a marker which has to do with vaccine, because it's inducing antibody response. Whereas the autoreactive cells responding to autoantigen lack this strong IL-21 cluster and also have other differences. So basically, this shows that the response to a vaccine antigen is different from the autoreactive response. Taken together, we show that we can distinguish at very early age children that progress and do not progress using this technique, and that in the future we will also be able to better characterize different cell types responding to different antigens by these ways. And finally, I want to thank the members of the lab and Ezio Bonifacio in Dresden, and also all the people in Munich, which have been very helpful for the sample collection and are managing all this children's data. Thank you very much.
Thank you. Time for one question or something before the wine and cheese? Or do you want to ask over the wine? I will ask when we go for the wine.
Okay.
Okay.